Hello everyone, I'm Dr.R.Vasuki, Professor and Head Department of Biomedical Engineering, School of Bioengineering, Bharat Institute of Science and Technology coming under Bharat Institute of Higher Education and Research. The course name is Biological Effects of Radiation. The course code is Bbm052. The topic is Biological Effects of Ionizing Radiation. Coming to the effects of radiation on cell at the molecular level. When radiation interacts with target atoms, energy is deposited, resulting ionization or excitation. The absorption of energy from ionizing radiation produces damage to molecules by direct and indirect actions. For direct action, the damage occurs as a result of ionization of the atoms on the key molecules in the biological system. This causes inactivation or functional alteration of the molecule. Indirect action involves the production of reactive free radicals whose toxic damage on the key molecule results in a biological effect. So you know what is a direct action and indirect action. The direct action means the radiation directly hits the DNA. The indirect action means it hits the molecules, water molecules surrounding the DNA. It forms a free radical, hydroxy radicals. And these free radicals, which are dangerous, it goes and hits the double stranded DNA. It is going to cause the damage, whether it may be a single stranded breakage or maybe a double stranded breakage, or it can remove the base from the DNA, which is very fatal in any case. Coming to the direct effects. Before going to the direct effects, you should know the atoms consist of smaller particles as protons, which are positively charged, neutrons, which are not having any charge, and electrons, which are rotating in an orbit around the nucleus of the atom, which are negatively charged. So what is radioactivity? If a nucleus is unstable for any reason, it emits and they absorbs the particles. So ionization is produced by unstable atoms. They are said to be radioactive. In order to reach its stability, the atoms give off the excess energy. This excitation, this we call it as the excitation, that is a radiation. This emission of energy, we call it as the radiation. So ionization radiations of the type alpha, beta particle, gamma rays, X-rays, neutrons, and coming to the non-ionizing radiation, with visible light or IR, microwaves, laser, a part of the ultraviolet rays, visible light, etc. So coming to the direct effect. Direct ionization of atoms in molecules, it is the result of absorption of energy by photoelectric and Crompton interaction. Ionization occurs at all radiation qualities, but is the predominant cause of damage in reactions involving high LET radiations. Absorption of energy sufficient to remove an electron can result in bond breaks. So this is the ionizing radiation. You can see that the ionizing radiation RH, it gives R minus plus. That is free radicals. So indirect action. These are effects mediated by the free radicals. A free radical is an electrically neutral atom with an unshared electron in the orbital position. The radical is electrophilic and it is highly reactive. So it is very, very dangerous. Free radical is very, very dangerous because it is going to hit the other atoms and it absorbs the electron so that it will become stable, whereas it is making the other atom as a free radical again. So this is a chain of reaction. Since the predominant molecule in biological system is water, it is usually the intermediary of the radical formation and propagation. So this is the indirect action. So indirect action, that is radiolysis of water. Free radicals readily recombine to electronic and orbital neutrality. However, when many exist as a high radiation fluence, orbital neutrality can be achieved by hydrogen radical dimerization. That is, we say it as H2 or H2O2. That is hydrogen peroxide. That is a formation of toxic hydrogen peroxide. You can see here in the down of the slide, in the bottom of the slide. So you can see how the ionization occurs in the water. HOH, the ionization goes to H plus plus OH minus. And this HOH, it is giving a free radical. H naught, you can see one O on the top. So that we call it as, the dot is called it as a radical, plus OH radical, which is very, very dangerous. So the hydrogen radical dimerization, the formation of toxic hydrogen peroxide, that is H2O2. The radical can also be transferred to an organic molecule in the cell. And coming to the indirect action, you can see the hydrogen radical and the hydroxyl radical, which we saw it in the previous slide. It is recombined to form the water molecule. And whereas a two hydrogen free radical will combine to form a dimer H2 or a peroxide dimer H2O2. How it forms? By the combination of two hydroxyl radical OH dot. So OH, or it can even combine with a neutral molecule to form a, it will transfer that radical. You can see that RH, the free radical is being transferred to the R. So it becomes R plus HOH. The presence of the dissolved oxygen can modify the reaction by enabling the creation of other free radical species with greater stability and lifetimes. So this hydrogen radical, when it combines with oxygen, it forms hydroperoxy free radical HO2 dot. And this organic peroxy free radical or not plus O2, it forms the organic peroxy free radical or O2 dot. Indirect action, the lifetimes of the free radicals. The lifetimes of simple free radical, whether it is hydrogen radical or a hydroxyl radical, are very short, on the order of 10 to the power minus 10 seconds. While generally highly reactive, they do not exist long enough to migrate from the site of formation to the cell nucleus. However, the oxygen-derived species, such as hydroperoxy free radical, does not readily recombine into a neutral form. These more stable forms have a lifetime long enough to migrate to the nucleus where serious damage can occur. Apart from this, I want to tell you one more radiation that is called as Bremsstrahlung radiation. What is Bremsstrahlung radiation? When high-speed electrons, when it may lose the energy in the form of X-rays, when they quickly decelerate upon striking with a heavy material, that we call it as a Bremsstrahlung radiation. I'll repeat it once again. Yeah, the high-speed electrons may lose energy in the form of X-rays when they strike a heavy material. So this we call it as a Bremsstrahlung radiation. I'll come to the radiation safety afterwards. How will you protect from all this radiation? By increasing the, by decreasing the time, by increasing the distance, and also by protecting yourself from the, by wearing a shield, aluminum shield or a lead shield. You have to increase the distance from the source of radiation, or you have to decrease the time. For example, if a person is employed in an X-ray room, he has to decrease the time he spends in the X-ray room. So thereby the contact, the radiation source, it is getting minimized. Or you just shift the people, no? Like day shift, night shift, or something like that. You have to give in a shift, the person have to be employed so that the same person will not be exposed to the radiation all the time. So this is the way it goes. Indirect action free radicals. The transfer of the free radical to a biologic molecule can be sufficiently damaging to cause bond breakage or inactivation of key functions. The organic peroxy free radical can transfer the radical from molecule to molecule, causing damage at each counter. Thus, a cumulative effect can occur greater than a single ionization or broken bond. So this is about the free radicals of indirect action. Coming to the biochemical reactions with ionizing radiation, you can see in the left side of the slide, the figure of the DNA structure. You can see how the ladder on the both sides by phosphate sugar, and in the how it is connected. Both the strands are connected by the ATGC base pair. This is the Watson Crick DNA base model. So DNA is the most important material. DNA structure we have been studying from the 9th standard, even from 6th standard children are studying now. So this is structure of the DNA. It is a double-stranded DNA, and it is held like this. It is a helix, and this is held by the hydrogen bonds between ATGC. So this is the most important material making up the chromosomes and serves as the master blueprint of the cell. So it determines what type of RNA are produced, which in turn determines the types of protein that are produced. So it is having a definite sequence. This DNA is having a definite sequence. So that particular gene has to be transcribed to a particular RNA, and this messenger RNA, and this has to be translated to a particular protein. Suppose if there is any mutation in the DNA, what happens? If there is any mutation in a particular gene, if there is any mismatching of the bearer a base, or the base has been replaced by some other base, then that particular RNA, mRNA cannot be formed in that same sequence, nucleotide sequence. So what happens when it is translated? The protein which is supposed to be formed in an appropriate manner will not be formed. So the different combination of amino acids. So what will happen if that particular protein, it is a hormone, what happens if, or an enzyme, so it may not be able to do the particular action. So this is what is diabetes. No, diabetes, the pancreas is secreting insulin, and that insulin hormone, if it is not having that particular amino acid sequence, it cannot function properly, it cannot bond bind, it cannot reduce the blood sugar level. So this is how the DNA, it has to be very specific in its nature. No mutation is allowed, no compromise at all. So the DNA molecule takes the form of a twisted ladder or double helix. The sides of the ladder are strands of alternating sugar and phosphate groups. Branching off from each sugar group is one of the four nitrogenous bases, cytosine, thymine, adenine, and guanine. So it determines what types of RNA, I'm repeating once again, what types of RNA are produced, that is mRNA, messenger RNA, we say. So it depends on the sequence, okay? The sequence of the mRNA, it is going to determine what is the sequence of the amino acids of the protein which has to be formed, which has to be produced to do its particular action. Biochemical reactions with ionizing radiation, that is DNA damage. There is considerable evidence suggesting that DNA is a primary target for cell damage from ionizing radiation. That's why we say that ionizing radiation is very, very dangerous, and pregnant women are not supposed to be exposed to radiation, especially X-rays. They should not be exposed to X-rays because it is going to affect the growing tissues, growing cells. Okay, the fetus will be affected. At the same time, growing children, they are not supposed to be exposed to the radiation at all. So fast growing cells, those tissues are very prone to damage by the radiation. So toxic effects at low to moderate doses, that is cell killing, mutagenesis, and malignant transformation, they appear to result from damage to cellular DNA. Thus, ionizing radiation is classical genotoxic agent. Biochemical reactions with ionizing radiation, DNA damage, same thing, it is a continuation. The lethal and the mutagenic effects of moderate doses of radiation result primarily from damage to cellular DNA. Although radiation can induce a variety of DNA lesions, including specific base damage, it has long been assumed that unrejoined double DNA strand breaks are of primary importance in its cytotoxic effects in mammalian cells. So that's what I told you, this ionizing radiation, or in case it is direct or indirect, if it is ionizing radiation, either the radiation strikes directly to the DNA, it is going to break the strands, or break the double strands, or it may hit the single strand. There may be a single strand break or double stranded break, or it may even remove the base pairs. So this is why this is very, very dangerous. So active enzymatic repair process exists for the repair of both the DNA base damage and strand breaks. In many cases, breaks in the double-stranded DNA can be repaired by the enzymes DNA polymerase and DNA ligase. So DNA ligase is the one which connects the broken ends. So our body itself, the defense mechanism, we are having a defense mechanism which consists of many enzymes which corrects this damage which is done by the radiation. But if it goes beyond that, this defense mechanism cannot work out. So the repair of double strand breaks is a complex process involving recombinational events depending upon the nature of the initial break. Residual unrejoined double strand breaks are lethal to the cell, whereas incorrectly recoined breaks may produce important mutagenic lesions. In many cases, this DNA misrepair apparently leads to DNA deletions and rearrangements. Such large-scale changes in DNA structure, they are characteristics of most radiation-induced mutations. Radiation-induced chromosome damage. Chromosomes are composed of deoxyribonucleic acids, that is DNA, which is a macromolecule containing genetic information, so which passes from generation to generation. This large, tightly coiled double-stranded molecule, it is sensitive to radiation damage. Radiation effects range from complete breaks of the nucleotide chains of the DNA to point mutations. That's what I said, point mutation, it removes the base, to point mutations, which are essentially radiation-induced chemical changes in the nucleotides, which may not affect the integrity of the basic structure. After irradiation, chromosomes may appear to be sticky with the formation of temporary or permanent interchromosomal bridges, preventing normal chromosome separation during mitosis and transcription of the genetic information. So transcription means the DNA is converted to RNA. So in addition, radiation can cause structural aberrations with pieces of the chromosomes break and form aberrant shapes. Unequal division of nuclear chromatin material between the daughter cells may result in production of non-viable abnormal nuclei. The chromosomes are composed of deoxyribonucleic acids and macromolecule containing genetic material. Again, I'm repeating. So this large, tightly coiled double-stranded molecule, it is sensitive to radiation damage. Radiation effects range from complete breaks of the nucleotide chain of the DNA to point mutations, which are essential radiation-induced chemical changes in the nucleotides, which may not affect the integrity of the basic structure. So this is the structure of the DNA. You can see the single-stranded break, and see, you can see here there is a single-stranded break. If both the sides, if both the strands breaks, then it becomes a double-stranded break. Maybe sometimes a single-strand breaks, maybe a DNA ligase, DNA polymerase, the defense mechanism can make it correct. But if double-stranded breaks occurs, then it is a very fatal thing. And you can see here a basic site. What is A basic site? These are all the ATGC base pairs. You can see how the two strands are kept intact by the hydrogen bonds, AT and GC. You can see here the radiation strikes this, we call it as a point mutation. How a base has been removed. So the base is missing in that place. So we call it as a basic site. So here you can see that there is an alteration in the base. So you can see the ATGC pair, that there is a different alteration. There is a different structural alteration which is done by the radiation. So you can see there is a base alteration which is not correct. When it is transcribed, it has not been transcribed to the correct mRNA, even after the single-stranded repair. So so this is how when radiation, that is the photon light, whether it is an X-ray or whatever it is. So when the radiation strikes the double-stranded DNA, you can see how a single-stranded break forms. This we call it as the direct radiation. And coming to the indirect radiation, you can see the photon. It strikes the water molecules which surrounds the DNA molecule. It is not striking directly to the DNA. So that is the direct effect. So this is an indirect effect. So it causes radiation of the water molecules surrounding the DNA. It forms the hydroxyl radical, and this free radical, it goes and hits the DNA. So this is an indirect action. So I'll repeat this once again. This is the direct action. That is the radiation photon directly hits the DNA. It causes a single-stranded break, and it's going to do its damage. This is the direct effect of the radiation. What is indirect damage? The radiation is going to strike the water molecule which surrounds the DNA, and this water molecule, it is undergoing ionization. It is going to cause the free radical formation, and this free radical, hydroxy radical, it is going to hit the strands of the DNA. So it is going to cause a damage to the DNA, but it is an indirect way. It is not directly hitting as in the first case. So this we call it as the direct effect, and this we call it as the indirect effect of radiation. So this, as I told you before, a simple double strand break, you can see here, which is a complex lesion, which is very difficult to repair. So both the strands are broken by the radiation. What is chromosomal deletion? You can see here, there is a chromosome. You can see that center of the chromosome, you have a centromere, and the ionization is hitting like this. It is the radiation strikes the chromosome. It breaks in the two sides. It is breaking. So you can see that you can see afterwards this broken part, it combines together and it forms an acentric fragment. What does acentric fragment mean? A fragment without the centromere, you can see here. Whereas the centromere is missing, whereas the last tail end, it goes and connect to the broken end, and it is forming the chromosome with a with a short of a with a few length short because it has been broken. It is forming a acentric fragment. So this we call it as a chromosomal deletion. A part of the chromosome has been deleted. It is forming an acentric fragment because there are two cuts in 2 points. The radiation is cutting the chromosome, which is having a centromere, so that one will not have a centromere. So that we call it as a acentric fragment. After this damage goes off, you can see that the tail part will come and connect to the broken end of the chromosome. So it is forming a chromosome with the centromere, but with a part of the chromosome deleted. And this is forming an acentric fragment. What is acentric fragment? A fragment without a centromere. That we call it as the acentric fragment. So which is of no use at all. So first we have seen deletion, how it is deleted. Now we are going to see translocation. You can see there are two chromosomes here with a centromere. You can see that the radiation strikes both the chromosomes, and how it forms. Just see how there is a translocation of the cut end. See this part, that red part of the chromosome, you can see that it is joining with the blue one, and the blue cut part, it is joining with the red one. That is translocation of the deleted chromosomes. So this we call it as the chromosomal translocations. So this is the original microscopic structure of the DNA. You can see that chromosomal translocation, chromosomal deletions. You can see it here. And this is the structure of the normal tissue. You can see that this is the differentiated cell layer. This is the dividing cell in the basal layer, and this is the basal lamina. You can see in the metastasis, this whole thing, this dividing cell basal layer, it will be multiplied. That is causing cancer on radiation. So coming to the cell initiation, an initiating event creates a mutation in one of the basal cells. You can see that when the radiation strikes, it is causing the mutation in one of the cell. You can see very few. One of the cell, it is causing the mutation. Dysplasia. So now what happens? This will initiate the mutations in many cells. The more mutations occur, the initiated cell is going to so it will proliferate. You can see how in the previous slide you could see that how it was only 2 layers. Now it will proliferate. You can see how many layers are formed. So this is dysplasia. Rapidly dividing cells begin to accumulate within the epithelium. So this is the initiated cell is going to proliferate at quantity. So still it is going to proliferate. You can see in the next slide. So you can see how it has been proliferated. So more, this is a benign tumor. So more changes within the proliferative cell line led to full tumor development. And this is malignant tumor. The tumor breaks through the basal lamin. The cells are irregularly shaped and the cell line is immortal. They have an increased mobility and invasiveness. So metastasis. So you could see how we have seen in the previous only 2 layers. See how the cells have been proliferated, abnormal growth. So cancer cells break through the walls of the lymphatic vessel or blood capillary. They can now migrate throughout the body and potentially see new tumors. So how you can see how the cells it breaks the wall and it is circulated in the blood, and it moves to the other parts of the body, and it initiates the tumor. So how we have seen how cancer, tumor, metastasis, all the stages we have seen. So radiation induced membrane damage. Biological membranes serve as highly specific mediators between the cell or its organelles and the environment. So that is the role of the biological membrane, highly specific mediator between the cell and the environment. So alterations in the protein that form the part of the membrane structure, because protein is very important for the susceptibility of the membrane, biological membrane. So if there is any leakage, suppose you just imagine a balloon filled with ink or any color. So what happens if you prick it with a pin? What happens? That blue solution it's going to come out of the balloon. So the same way, biological membrane, they have to be very intact, susceptible to any damage. So alterations in the protein that form part of the membrane structure can cause the changes in its permeability to various molecules. That is electrolytes. So what happens if there is any alteration? The permeability of the membrane changes, so it will allow all the electrolytes to move both the sides. So in the case of nerve cells, this would affect their ability to conduct the electrical impulses. So electrical impulses is a vital for nerve cells. So what happens? So in the case of lysosomes, the unregulated release of its catabolic enzymes in the cell could be disastrous. So if there is any damage, and we say that, the heart damage or a liver damage, the particular marker enzymes which will be stored there, it will be leaked into the, if there is any membrane damage, if there is any cardiac tissue damage, what happens? That particular SGOT, we call it as SGOT, glutamate oxidase, what happens? It will be thrown into the bloodstream. So if that level is found to be increased, that is an indication that there is something wrong with the cardiac tissue. Okay? So in the case of lysosome, same way, the unregulated release of its catabolic enzymes into the cell could be disastrous. Ionizing radiation has been suggested as playing a role in plasma membrane damage, which may be an important factor in the interface, that is the cell death. Coming to the cell cycle, undifferentiated cells that are growing or destined to divide. The generation time from one cell division to the next, it is known as the cell cycle. It is independent on species, tissue type, age, and environmental influences. Mitosis occurs with the division of the cell. The cell cycle can be divided into two phases: G1 gap, S synthesis, G2 gap, and M, that is mitosis. Cells not actively growing occupy a fifth phase that is known as G0. The cell in G0 can often be stimulated to enter the active cycle by environmental stresses. Cells are most sensitive to cell killing during the period shortly before M phase, at late interphase, that is G2, and during M phase. Higher resistance is seen in cells in S phase and late G1 phase, as well as all cells in G0 phase. Resistance in S phase may be due to the presence of synthetic enzymes capable of prompt repair of DNA breaks. However, mutation frequently increases in the cells in or just before the S phase. Irradiation of the cell causes cell death at mitosis as a result of inability to divide. That we call it as the mitotic death. RNA and protein synthesis do not halt in the sterilized cell. The result is a production of the giant cell whose unbalanced growth eventually produces lethal to the cell. Bergoni-Tribonde law. See, according to this law, early radiobiologist in 1906, radiation response in tissue with a function of a high number of undifferentiated cells in the tissue, a high number of actively mitotic cells, the length of the time the cells remain in active proliferation. So the radiation response in tissue is a function of 3 points. One is, the first point is a high number of undifferentiated cells in the tissue. There is a high number of undifferentiated cells. Next is high number of actively mitotic cells. Last one, the length of the time the cell remain in the active proliferation. Radio sensitivity. It is not clear why lack of differentiation of the cell results in radio sensitivity. It has been shown that undifferentiated cells or cells in the process of differentiation are easily killed by radiation. So the length of the time that cells remain in active proliferation relates to the number of divisions between the most immature stage and the final mature stage. The longer the cell remains in active proliferation, the greater sensitivity to radiation. That's what certain particular organs are very prone to radiation damage, whereas like muscles or brain cells, they are not affected by the radiation because not much proliferation takes place here, not much growth of the cells there. Thank you.